interlude there. I would like to invite everybody or welcome everybody to can you see my screen?
Yes.
Okay, the first in our new series of botany speakers, in particular early career botany speakers, with a focus on graduate students and recent graduates, such as Dr. Villanueva. And this is the list of upcoming speakers. So we have one a month. I'm actually really super excited to hear many of these people. I've never heard them speak before, and learn all about what various people who either are Western botanists or were and will be again Western botanists have been up to. Um, in the meantime, let me introduce today's speaker, Lorena, who graduated with her PhD from UCR a few years ago, 3?
It almost feels like a few years ago, but no, it was 2019.
Okay, 2020 was long enough for years. So since then, so Lorena is particularly interested in outreach and science writing. So since she left, she was an intern in the education department at the National Zoo at the Smithsonian. She was public engagement officer at the U.S. Botanic Garden. She was a AAAS Mass Media Fellow at the Indianapolis Star and spent some time in Indianapolis for that. And she's now a freelance science writer and editor who's had pieces in Science, the ASPB newsletter, and Mexican outlets such as Este País and Capital. And she was also the outreach coordinator for the California Botanical Society. And probably, I know while she was at UCR, she did a lot of stuff, but in the interest of keeping this short, I will leave it at that and say thank you very much, Lorena. And it's up to you now.
Wonderful. I'm going to start sharing my screen. Thank you for that introduction, Amy. And this takes a few seconds because it's a really heavy presentation. Not to fret, though. I'm going to make it as interesting as possible. I really want to thank you all for being here, sharing your time with me, giving me your time. I know that I'm in Mexico City and you could probably be watching TV. My mother is watching TV. I'm at her house right now. So I deeply appreciate it. I am going to be talking about my PhD project, but also of the reasons that made me decide to take on this PhD project and the reasons that I keep working in science and science communication. So forgive me if I go a little bit into the personal story behind all of this project. But I think it's also important, given the times that we're living, to remind ourselves and to remind our colleagues of what makes us passionate in life. So I would also like to thank, of course, the funding sources, because nowadays I realize more and more how important money is in order to realize your dreams and to travel to the places that need to be studied. And so I would like to thank my funding sources, CONACYT and UCMEX is, of course, my home institution. The institutions and offices in the US that gave us grant permits to collect in the US. And also a group of colleagues and friends that helped me throughout this process. Of course, my advisor, my co-authors, people in my lab, people that I have mentored, and friends that have helped me collect all these beautiful leaves that I will be telling you about, and of course, the rancheros in Baja California. So I'm going to start with this personal story of why I decided to do what I did for my PhD. I traveled to San Jose del Cabo, which is at the tip of the Baja Peninsula, in 2006. I was quite reluctant to do this trip because I was not very keen in seeing my dad at that point, but my mom insisted, very fortunately. And so together we traveled to places that were close to the Cape region. And here I am in what is now the Marina of San Jose. This, of course, is almost an unrecognizable site because of the Marina itself. And during that trip, I made a really important decision because of what you will see here. These photographs are from various places throughout the peninsula that were given to me by Ben Wilder. I have a few of mine there. But this is just to show you that the peninsula, as many of you might know, is just beautiful. And even saying beautiful is an understatement. It just bewitches you, and you have this longing to always go back and to protect these places. So in 2006, I realized that the peninsula was in danger, and this was only the Cape region. I was quite surprised at the number of hotels that were being built. And instead of pursuing a career in Hispanic literature, which was my original plan, I decided to become a biologist and to collect data to protect the places that I care about the most. The current times that we're living in evidence that data might not be enough, right? So that's another part of the talk that I will focus on, which is telling stories. However, data was important because, as you know, protesting in Mexico is not very efficient. And at this point, many of my father's friends were protesting in Highway number 1 against the construction of these hotels. And I thought, no, data has to be the way. I have to publish some papers. And so the way to do that was to contact someone who I consider the superhero of conservation in Mexico, who was my advisor, Dr. Ezequiel Escurra. And together, we designed this project that I will be talking about. We went to the field together. I don't think that he really likes to be photographed. So that's why you see him really tiny in that little picture. And although it was not my intention to go to the US, I moved to Riverside for my PhD. And very quickly, I learned about the history of the place. I started looking at the plants that surrounded my environments, and Washingtonia is a very obvious one in California, and especially in Southern California. And one of my interests is also history. And throughout the course of this investigation, I found amazing historical photographs that really sparked my interest in this group of plants. So this is a series of photographs of Magnolia Avenue, an important avenue in Riverside. And you can just see that during the late 19th century, palms were pretty much shaping the cultural identity of California. I can totally identify with Charles Nordhoff in how he was promoting the settling in Southern California at the time. He was writing that California is our own, and it is the first tropical land which our race has thoroughly mastered and made itself at home in. There and there only on this planet, the traveler and resident may enjoy the delights of the tropics without their penalties, a mild climate, not enervating, but healthful and health restoring. And this is just a slide to show exactly what he was referring to and what we see now. In LA, the LA Times published in 1886 that the enormous number of palms planted in Southern California during the last 3 years will ultimately tend to give a really tropical appearance to our towns and villages. And if you look to the image on the right of your screen, the license plates of California, that was 2012. And you can see the really tall and skinny Washingtonia Robustas on the left. And on the top, you see this, the Mission San Fernando in the semi-tropic California publication that is Washingtonia Felipera. Now, one of the aspects of this investigation that I also found fascinating was the taxonomic mystery itself. So when I finished doing my undergrad in biology at UNAM in Mexico City, I became a plant taxonomist. So it's something that you can hardly shake off. So the minute that I came across the name Washingtonia Robusta, I went into Tropicos, wanted to learn about the type, wanted to learn about the original publication. And this is the type for Washingtonia Robusta. Of course, this is not the ideal type because it's a young specimen. We don't get to see any of the diagnostic traits that could be useful in distinguishing it from Felipera. And this is the original, or part of the original description, the prologue. The plant was first described by a German botanist who never visited America, so he was not the person to collect the plant himself. And he thought, from what other people had told him at the moment, that the species originated from the Sacramento River in California. This, of course, we now know is not true. However, the question remained, where was Washingtonia robusta originally collected? This illustration is from 1885 and was designated as a type in 1999 by Richard Felger. The illustration is from a young specimen, as I had mentioned, cultivated in the south of France. Remember, at this point, it's not only California that is being palmerized, if I should use that verb, or is being inundated by these palms. It's all around the world. Palms are, at this point, a symbol of status and of wealth. And people were pressing Wendland to tell them where he had or where his collectors had collected the seeds. So in 1888, he had to recognize that he did not know the original place where Robusta had been. Now, of course, this sparked even more my interest. William Greenwood Wright was a natural historian of Southern California at that time. And as a scientist, he was interested not on the collecting side of it, but he was interested in defining the species of Washingtonia robusta. This letter very much illustrates that. In 1879, he wrote that he would take to the field in person and cut down 100 palm trees if necessary to settle the matter. So I thought to myself, if this person is as intrigued in 1879, why isn't this solved? Or why is it not really solved? In 1889, he wrote to Samuel Parrish. He wrote about Samuel Parrish that they both believed that Robusta was a myth and a fraud. So for me, those words were really incendiary. And I made the purpose of just compiling this information to make sure that what had been normally used as a taxonomic character, which is a stem diameter, was really a useful trait. So I'm going to show you the part in the upper side of that table. Hermann Wendland, who was a German botanist that described both species, he did not give any measurements, neither for Robusta and for Felipera, and that's pretty understandable because the plants at that point were super young and there was no stem to measure. So that makes a lot of sense. And you will notice that it was not until 1936 that Liberty Hyde Bailey gave some measurements, but it was for Washingtonia Felipera, not for Robusta. And until 1999, we had some type of measurement for Robusta. That was because of the work of Richard Felger and Elaine Joyal in Sonora. But then again, you see that the measurements are pretty inconsistent from then onwards. Scott Zona in his treatment of the Aracaceae for the flora of North America. And then lastly, Scott Simono in 2012 for the Jepson manual considered that any palm that had less than 0.8 meters in diameter could be considered Washingtonia robusta. So this is exactly how the palms from the edges of the distribution look like and what we normally see in the streets of Southern California. This is Riverside. And on one side of the street, you will see Washingtonia Felipera, and then on the other, you will see Washingtonia robusta. The natural distribution, you can see on the right. Felipera occupying the northern oases, corresponding to Southern California, Northern Baja California, and then one site in Arizona, which is found in the Cofa Mountains, and then Washingtonia robusta extending from the Cape region until, and this is where things vary between authors. It can go up until, or used to go up until the dash line A or the dash line B. But the interesting part is that area between both dash lines that remained as a big question for taxonomists that had been looking at Washingtonia. And therefore, what I wanted to answer with my PhD was first, see how vegetative morphology was varying in natural populations along its distribution range. And I'm focusing on vegetative morphology because these are the traits that have been used consistently or in the traditional literature of Washingtonia to distinguish between both species. And in order to explain the variation that we see in Washingtonia, two hypotheses can be invoked. The first one being the hybrid zone model that posits that there's two species of Washingtonia, one found in the south and then the other one found in the north, that are connected by a hybrid zone. On the other hand, we could explain this variation by recurring to the Klein model hypotheses, in which we could consider each of the populations varying very gradually along the latitudinal Klein or along the geographical gradient throughout the peninsula. Now, the Klein is not a new concept, of course. It was first proposed in 1938 by Julian S. Huxley to refer to a geographical gradient in phenotypic characters, meaning that each of the populations that precedes the one before is changing very gradually in some of the traits. In this case, I was analyzing them in morphological traits. It has been used to study these other groups of plants, and it could be interesting to examine the genus Brahea and Pachyserius just because they show very similar patterns to the ones that we found in Washingtonia. And therefore, we sampled the oases. Of course, these are not the only oases of Washingtonia that are found in Mexico and the US, but this is a very exhaustive sampling that we did, and that represents the geographical extent of the genus. This is one slide that I really wanted to show at some point, and this is just the exact time to do this. People oftentimes had asked me in conferences or in other presentations why we didn't collect more data, why we didn't put some sensors, or why we didn't collect any flowers. We only have one specimen that had flowers and they were old. And I always used to say, well, it's because it's a remote and inaccessible place. This is the Rendo Canyon in Mexico. And the first time that we went there, I hope you can see the face of my lab mate, Andrew. We got stuck. We were not using a 4x4 vehicle, and this was June. Very responsibly from my part, I have to say. We got stuck and we were just shoveling. It took us 2 hours to get the truck out. At that point, we had already walked 8 kilometers in and 8 kilometers out. It was an ordeal. But I had a really great crew that helped me all throughout the process. The next picture that you're seeing there are 2 students from Sonora, the University of Sonora, that were helping me collect the ponds in Nacapule Canyon. And then my 2 friends, Elenice and Antonio, who made really good use of the Jeep, just to use it as a ladder. This is Sierra Cacachilas in the state of Baja California Sur. And in each of these sites, we collected morphological data of 20 individuals, the 10 tallest individuals, and then the individuals that we could reach however we could that would measure between 5 and 7 meters. And the traits that we took were height, crown width, diameter at breast height, and diameter at base. Remember, because these 2 traits were important to test if they could hold if they were really reliable characters. And from those individuals that we could reach, and I'm telling you, these plants can be massive. That's a friend of mine, Eric. He's 2 meters tall, and that's a palm in the Rendo Canyon, which is huge. We also collected leaves. We measured the length, the width, the petiole width, that pastular length. And then because in my team, I was very fortunate to have an ecophysiologist, we took little fragments of leaves for specific leaf area. We took photographs to measure the colors of the leaves, because in some of the publications of Washingtonia, Liberty Hyde Bailey, for example, or Samuel Parrish had spoken about the differences in color between the phenotypes in the north and then the phenotypes in the south. And then also measurements of stomatal density and delta carbon 13. What we then did is we ran a simple linear model with latitude and site as predictors of each of the traits. And what we found was that most of the traits, the ones that you're seeing highlighted in green, are correlated with latitude. However, what is also interesting to see is that the site also is playing a big part in the variation of these traits, for the exception of adaxial stomatal density, which of course is not surprising because the environmental conditions in each of the canyons is very variable. However, we started figuring out that there might be a really important interplay between environment and genetics, as some of the horticultural transplants showed. So on the left, you're seeing a photograph of a visit that Kate Sessions made with T.S. Brandegee to the Cape. At that point, she collected seeds and then took them to San Diego. And there's a photograph of 1927 where the phenotype is not very clear. It could be Felipera or Robusta. But then my advisor took a photograph in 2018, and you could see that the phenotype of the Cape region is still maintained. What we then did is we took the latitude correlated traits and we ran a principal components analysis, and these are the results of that. So what you're seeing is things that are not really surprising. We're just confirming what others had observed before. In Baja California Sur, which is close to the Cape, we're seeing these populations where the palms are very slender, have thin and green leaves. And then we have the northern phenotype, which would correspond to Washingtonia Felipera from Palomar Canyon in Baja California, in the state of Baja California in Mexico, Joshua Tree, Palm Canyon, and Hellhole Canyon in Anza Borrego. However, we still see that that area that I showed you in the map in the beginning is still a question mark because neither of these canyons are lining up with either of the phenotypes. There's also uncertainty from this PCA of the identity of the palms in Arizona and then the identity in Sonora. We then took the first principal component and ran a linear regression against latitude. And what we found is that COFA was an outlier of the otherwise continuous trend of the peninsular California populations. Then we adjusted the hybrid zone, the logistic model, to test the hypotheses of the hybrid zone and compared both of the models and decided for the Klein model based on information criteria theory as well as with the F value. So what this shows us is that, of course, the Klein model is providing a better fit to the data. And biologically, what this means is that Washingtonia is composed of one highly variable species, meaning that it's changing as one moves towards the north. We wanted to confirm our results now using genetic data. I was personally interested in figuring out whether there was another level of resolution that could be possible with the new technologies. And for this, I'm going back to this personal story. One day I was riding the bus going to UCR, and a friend of mine was on the bus, and I was telling him about this problem that I had. This was Jacob Landis, and Jacob said, well, we could work on this together. Why don't you talk to my PI? And that's what we did. And so we're all co-authors of that publication. And in this case, we wanted to see whether the SNPs were revealing similar patterns to those of the morphological traits, and what was the population structure as well as the phylogenetics relationships of Washingtonia. In order to do this, then we performed genotyping by sequencing on 188 Washingtonia individuals. We had already collected the leaves and they were sitting in the lab. And then we analyzed the data using two different approaches. First, the de novo approach and then the reference-based approach. We used both to confirm our results, but also just to make sure that the reference-based approach was not too far off, because Phoenix dactylifera is not a close relative of Washingtonia. And what we found in this base factor species delimitation was support for 3 subspecies. In green, you will see the populations in the south of the peninsula, and in blue, those in the middle of the peninsula, and then finally the ones in the north marked in red, pink. So this gives us support to 2 of the phenotypes that we had seen before that have been traditionally recognized, but also of the existence of a new subspecies or a new taxon found in the middle of the peninsula that had been a big question for taxonomists. We then did, again, the same approach that we had done with the morphological traits. We ran a principal components analysis, this time using SNPs. And what we saw with the first 2 components were that the populations were being separated south and north. But then very interestingly, when we examined the third axis, this third axis is explaining the SNPs variation or the unique genetic variation that corresponds to Sonora, to the population in mainland Mexico. We took the first principal component and we ran a model against latitude to test our both both of our models, the Klein model and then the hybrid zone model. And we scored the PC, well, we did a histogram of the PC scores, and we immediately saw that there was a mixture of distributions that when colored, we will see the green corresponds to the south of the Baja California Peninsula. The yellow one is Nacapule in mainland Mexico. In pink, it's San Ignacio, a very special population in the middle of the peninsula. In blue, the mid-peninsula region, and then finally in pink, the northern oases. Because we were really interested in understanding whether that mid-peninsula region was not a hybrid zone. We ran a structure analysis, and what you see is that each of the genetic regions that we found doesn't have any genetic influence from the other ones, for the exception of Alamos, which is marked in pink. It was a population that I was referring to previously. San Ignacio has a mixture of genetic influence from the mid-peninsula region and then the south of the peninsula from Mexico. And this can be explained after looking at this migration model, in which we see that genetic material was introduced deliberately, or what we presume to be deliberate, from the south of the peninsula in Mexico to Alamos, which is San Ignacio. We also were interested in understanding why we were seeing those genetic regions. And for that, we took a look at the literature for the vicariant events of the peninsula. So of course, it made sense that the Nacapule region held its own genetic diversity since the opening of the Gulf of California. Also, our results would confirm the existence of the seaway in the middle of the peninsula. However, we did not find any support for the inundation event that has been proposed for the Isthmus of La Paz. One thing that at least I am interested in understanding is what break, or what is the reason of the isolation of the populations in the northern part of the Baja Peninsula and the ones in Southern California. So what is making these populations from the mid-peninsula region and the northern peninsula region be so distinct? So just as a summary, what we found is, of course, the evidence of a northern genetic cluster, which would correspond to what we know as Washingtonia Felipera. Another confirmation of the existence of a phenotype and a genotype of Washingtonia Robusta in the south of the Baja Peninsula. And finally, just the evidence that we needed to show that there's a different genetic identity in the middle of the peninsula that is marked now in blue. So as a conclusion, of course, there's distinct genotypic regions along the latitudinal Klein. There was no hybrid zone, for the exception of that population, which was San Ignacio, and that the regions are coinciding very well with phylogeographic breaks, for the exception of the north, the separation between the north and the mid-peninsula region. And our genetic data was confirmed our morphological results, in the sense that palms are varying clinally. As I said, it would merit further investigation to use this Klein approach to understand the variation in the genus Braea and the genus Pachyserias, but also, quite interestingly, in a group of palms that inhabits in the southern hemisphere in Argentina and Brazil, which corresponds to the genus Tritrinax. Now I'm going to switch gears because it's something that I have been thinking about lately. And then, well, I had my data, I had my papers, which are not a lot, but I have my papers. And then I realized that data might not be enough. It's something that I had always figured out, or had always thought about, but I wanted to take a more serious step. And so I applied for a AAAS Mass Media Fellowship. And I was a fellow writing for the Indianapolis Star during the summer of 2020. I was really lucky that the fellowship didn't fall through. And one thing that I noticed while visiting one of the field sites for a story about bats in Plainfield, Indiana, was that beautiful places, of course I knew, but I just confirmed this, are everywhere. Baja is Baja and will always have a special place in my heart. But there's places like Plainfield, Indiana, and I was very happy to go with these researchers, not only because I was again confirming that beautiful places are everywhere. But because I was going with a group of students and researchers, and I was not in charge of leading the expeditions anymore, or of shoveling sand from underneath the truck, this is the group of researchers I was with. I also was accompanied by a photojournalist from the paper. She was amazing at just making people talk to her, and not necessarily scientists or not necessarily about science. And then you can see one of the students, she's counting the number of bats that are coming out of the bat boxes. And the principal investigator of this project, she's holding a microphone just to capture that sound of bats. I'm very happy to say that the story made it to the front page of the Indianapolis Star. I still keep in touch with the PI, and the PI was recently telling me that she has been contacted with people asking her what to do about bats that are living in their attics. So it just made me confirm that data, of course, as evidenced by these group of researchers, is necessary, but we need to get the message out. And so I don't know what's next for me, but at least I feel very happy with the things that I've done.
It's a lecture.
I know, as much as of a cliche as it might sound, I am sure that you will agree with me that no matter where the road leads, we're all in this to make this world a better place. So with that, I would like to really, really thank you for your time, for being here. And if you have any questions, I would be more than happy to answer them.
All right. Thank you, Lorena. That was really great, even if it was, what, the third time or something that I've heard this story, at least, but it still is just a fantastic story with really, really solid, great research. If you have a question and know how to use the raise your hand function, that would be super great. Or if you want to put a question in the chat, and if none of those work, you can try to just throw it out there, although most of you are muted, so you'll have to unmute yourself. Questions, anybody? I see people unmuting themselves.
Lorena,
Yes?
can you hear me?
Who is this?
This is Janet. I don't know. Can you hear me?
Yes, I can hear you.
Oh, okay. So I'm curious, is there just two native palms in Baja California, the Robusta and the Felipera? Or is there more than just the two?
Well, currently I'm working on the taxonomic treatment. It's going to be Washingtonia Felipera, subspecies Felipera, subspecies cataviñensis, and subspecies sonorae. So that would be the three subspecies for the peninsula as we stand right now in 2021.
Okay. All right. I'm familiar with the Felipera. I am in Anza-Borrego a lot, and I'm familiar with those palms, but I've never been familiar with the Robusta. So it's interesting to see how the migration and the how the Robusta is in the southern part of Baja, and then the Felipera is up north. So thank you. It was interesting.
Thank you for your question, Janet. Yeah, one thing that really surprises me is how well we know Robusta in the city and how little we know of Robusta in the field. One thing that I didn't mention during this talk is that it is considered in California, for example, an invasive species, Washingtonia robusta, and then in Mexico, we really don't have any idea of if it's facing any type of risk according to the IUCN. As I said, also, one thing that I'm just thinking right now is if there are any other canyons in which we would have the same genetic identity of Robusta, or that would correspond to Robusta that we still don't know about. So Robusta pretty much is still continuing to be a mystery. It's solved. That's something that I should say. It's solved because I have worked on it. But it's so poorly understood in the wild. So yeah, it's really ironic, really.
Arlie, I see your question, but I'm going to ask John Randall to ask his question.
Sure, it's essentially the same as Arlie's.
Okay.
Lorena, if you looked at any of the plants that are invasive here in Southern California, or it's actually invasive even further than Southern California, up into the Central Coast. Did you look at the genetics or the morphology of any of those? And they're often invasive, as you know, in the coastal zone, in the cities and in arroyos elsewhere.
In my garden.
Yeah, I didn't. One thing that is problematic with the genus, and I didn't want to complicate things further, was to separate those populations like the one in San Ignacio, which might be the result of human introduction, from the natural distribution. I think the first step towards the understanding of Washingtonia, which is actually the very same title that I think Liberty Hyde Baby used, was understanding its natural distribution and its natural variation. So no, I didn't include the ones in Southern California.
Yeah, I'm just fascinated by, so especially given a lot of proposals, not necessarily with the Washingtonias, but with a variety of plants that we feel like we may need to start moving north due to climate change. This one might be successful on its own, like it's already begun, maybe. So I'm just super curious about what the genetic origins of the invasive types are, what so-called invasive types are.
I would be as well, because I mean, this is not the place where I would like to say this, but if the populations were to be extirpated from some of the natural places, then at least we know that there's still some type of germplasm in Southern California.
Right, yeah.
Not that it's ideal, but there's something still left.
Yeah. If I may, I'll just offer a thought too, and this on something totally different. You asked, you left open that question of why the break between the northern type, which is in northwestern or northern Baja California state, and in Southern California, and the central type. And it looked to me like I'm looking at a very coarse map and big dots on that map, but it's like I noticed, gosh, that looks like San Quintin, immediately west of the southernmost northern type. And that's where we, just south of there, Santa El Rosario, think of the Mediterranean climate regime on the coast ending. And so it just sort of like struck me like, wow, that looks like the Mediterranean type up north, even though I know it grows on the desert side. So just a curiosity to me.
Yeah, that's interesting that you mentioned. One of the other things that I think would be good to know is the phenology. If these palms are flowering at the same time or not, because they're not that far apart for them not to be exchanging or not to have any gene flow. Yeah. Thank you for that.
So specifically, Arlie's question was, did you notice any difference in the invasiveness of Robusta and Felipera? And I'm going to guess that the answer there is no, since you didn't really look at that.
No, but one good clue to this, it's not something that we investigated specifically, but specifically Feria is a very good indicator of the behavior of plants. So plants that have high specific leaf area, as is Robusta, or as is the case in Robusta, are plants that have rapid growth rates. So I would imagine that that's a clue of how rapidly a palm can invade. They use the resources really fast, and they would use the resources in changing in the leaf changeover really quickly, as opposed to Felipera, in which the specific leaf area is really high, and these are plants that grow really slowly, that would use the resources to make leaves that are more durable. So I think that would be a clue of the strategy for these palms.
Awesome. We have a question from Stephen. How closely does the morphological Klein line up with genetic structure across wild occurrences of Robusta and Felipera? How closely do the two line up?
I would say that they're very close from, I think I showed that map. Well, that's exactly no, because not all of these populations we use for the analysis, but it pretty much lines up to with the map that you're seeing on the right. The map on the right would be the morphological results, and you can see our genetic results on the left. So they're pretty much lining up, for the exception of Nakapule that I've colored in yellow in the genetic map, and I've left in green in the morphological. One thing to remember though, now that I'm thinking about this, is the population in Sonora, we didn't have any color. If you remember, it was an open question mark, as well as the Cofa Mountains. What we see is that the Cofa Mountains are the same genetic identity of the northern populations, and that Sonora is not the same genetic identity as the Cape region. It's its own genetic identity. So very similar, for the exception of those two populations.
There's a comment from Matt, in case you're interested. I don't know if you have time to look at it, but I'll read it. Many of the naturalized individuals I see in urban Southern California are what were classically known as hybrids, Washingtonia ex filibusta. And then he has a link there. So if you want to check that out. Thank you, Matt.
Thank you.
Yeah, does anyone else have questions?
Hi, Wayne Farron. I was trying to send one, but it was easier to maybe explain.
Go for it.
Thank you. In the mid-1980s, Madronio published a paper suggesting that, I forget the author's name, that the spines on the petioles were an anti-herbivory response to large Pleistocene mammals, grazing or browsing pressure. This brings to mind the idea that what was the conditions under which Washingtonia once grew and evolved, and that might help explain the invasiveness in wetter, milder climates along the coast for some of it. But it also, I think, expands the imagination to think in terms of Washingtonias being browsed by tall camels and the like. I'm here in Las Vegas, and one of the largest Pleistocene mammal deposits is here at the Tule Springs Fossil Beds National Monument. So the mind wanders, and I think it's an exciting thing to think about what might have been and where it's going today. You know, what does it mean for the future, as the other commenters were saying. So mine's more of a comment and a and a what if, but thank you. I really appreciate what you had to say.
If it serves for something, we did collect observations, at least, of the presence of spines in Washingtonia. We didn't end up doing anything about them, just to confirm exactly that observation. I think the paper was from Cornet. And what we noticed is that taller palms, and this is of course just by looking with binoculars or by just collecting some of the tallest palms, are indeed, they indeed lack the spines on the petiole, and then the younger ones would have the spines, but it's something that I didn't go into in much detail, I have to say. Thank you.
And we have the citation for the paper in the chat now. Thank you again, Matt. If anyone wants to check that out. Other questions? Might as well just unmute yourself and go for it. Thank you, Deborah. Will this talk be available to watch again? I had a terrible breach of protocol in not telling everyone that it was being recorded. So if anybody does not want their little Zoom name or something to be visible, let me know, or if you don't have my email address, which is amy.lit@ucr.edu, or you can write to anybody in the board. But otherwise, I think I will try to see if we can make it available. So unless you have an objection, Lorena.
I'm totally fine.
I'll work on that, and it's one of the things, unfortunately, that we didn't really think through in advance. So thank you for bringing that up, Deborah. And as Fenerty family says, you can save the chat by pulling down on the 3 dots in the chat box. Oh, I see. There are the 3. Okay. Save chat. Yeah, there you go. So at the bottom right, if you click on those three dots, it'll say save chat, and then you can have the Madronio citation and also the link to the information about the hybrids.
Can you tell me again where that is?
Sorry?
Can you tell me again? I think I see now. Yeah.
Yeah, okay. Open up. So for people who didn't get that, if you go to the bottom of your screen and click on the icon that says chat, and that'll open up the chat box on the right. And then if you go to the bottom right-hand corner of the chat box, you'll see three little dots. And if you click on that, you should see the link. Okay, any other questions? You're getting some love in the comments, Lorena.
Thank you.
But I think if no one else has any questions, we can let Lorena go.
Thank you, Lorena. It was awesome.
You. Lluvia, nice to see you. Thanks for joining us.
Kate, hi everybody.
Hi, Sula. This is so nice. Well, thank you all. And thank you for the invitation.
Sula, look at all these people who I didn't even know were here. And I'd love
Yeah,
to.
me neither. That's so cool. It's one of the things that I miss about presenting in person. You never know who ends up looking at your talk.